My name is Jonathan Jones, and I work at the Sainsbury Lab here in Norwich, and I basically work on trying to understand how plants resist disease. But some of that knowledge that we obtain we can use to try and reduce crop losses.
And how do you define achievement?
How do I define achievement?
Yeah.
I think we're all asked to describe our impact as scientists to justify our BBSRC grants coming from the taxpayer. I think you make a difference if you contribute to a better understanding of biological mechanisms, and you also make an impact if some of that insight can be used to solve real-world problems. I don't really know what was expected of me, but I had an interest in science from quite a young age. I guess I was given lots of little books, The Wonder World of Do You Know, which I try and find in second-hand bookshops from time to time, books like that. And they just provided ways of thinking about things that gave you a little deeper insight into things than you had from talking to your mum.
You said in your article you knew you had to do something I think is useful.
Yeah. So
even at that age.
It was a habit, I would say, or it wasn't even something I questioned. It was you had to give something back. That this is an ethic that my mother imparted to me. So I'm not really anyone who knows me will know I'm not a particularly holier than thou person, but I'm glad that through the kind of work I've done, I feel I've helped improve our understanding of an important mechanism that plays a part in an important problem, and so yeah, I think that's a way of partially at least giving something back. I think actually, if you think back to the sort of movies of let's say depictions of Watson and Crick and a double helix. What's really different these days is that anything important maybe being worked on in dozens of labs, across the world, not just one or two, and so there's an element of competition, but also there's success has a lot to do with good networking with other colleagues interested in the same thing and building partnerships. But I would say, if I look sort of the big breakthrough in my field, or a very important big breakthrough in my field, was to actually clone the kinds of genes that confer resistance, and that Flor himself was studying in the 1940s. And so Flor's genes, actually the flax resistance genes to rust, were cloned by a team in Australia. We cloned genes for resistance in tomato to a leaf colonizing fungus, and other people cloned genes for resistance to bacteria and fungi. And what was very exciting in sort of like 1993, 1994 was to discover that actually all these people were calling each other up in hushed tones and say, "What's your one look like?" And so the structure of the encoded protein that we all independently had cloned did provide clues about how recognition occurred, and then to varying extents clues about how, upon recognition, defense is subsequently activated.
So you guys all independently cloned it.
Yeah. So
that was huge.
That was pretty exciting, and there was a flurry of papers in Science and big reviews in Science that I was a co-author on, and that was so yeah, we had this flush of success, like we've cloned all these genes. Now how the bloody hell do they work? So in a sense.